Hello friends, let us now study the second part of lecture on microencapsulation. In the earlier part of this topic, we discussed about the basic principles of microencapsulation, and we also studied the physical and physicochemical methods of microencapsulation. Now, in today's lecture, we will study the chemical methods of microencapsulation like solvent evaporation and polymerization, and later on, after discussing these chemical methods, we will talk about the applications of this technology in food processing or in different food products. The solvent evaporation methods includes 3 steps. First of all, there is a formation of solution or dispersion of the core material into an aqueous solvent phase. This is followed by emulsification of the core material into an aqueous phase containing a suitable stabilizer, and therefore, this results in the formation of a water in oil emulsion or oil in water emulsion, as the case may be. After this, in the third step, the solvent is removed from the dispersed phase either by extraction or evaporation, and this leads to precipitation and formation of microspheres. The polymerization process occurs in the continuous phase rather than on both sides of the interface between the core material and the continuous phase. The microcapsule formation process is conducted by the addition of oil in a melamine formaldehyde resin solution and a sonication process to emulsify the oil in the aqueous phase. The resin is then added into the emulsion under continuous stirring, and finally, the adjustment of the emulsion pH is done to the acidic range. After this, we can see that the microcapsule cell formation occurs due to the reaction of melamine with formaldehyde at the interface of oil droplets, producing a cross-linked film of melamine formaldehyde polymer, and this whole process of polymerization is schematically or pictorially presented in this figure. You can see that is the solution of melamine formaldehyde. It is a pre-condensate form and copolymer added in water, and it has a cross-linked polymer, and then oil is added step by step, all right, and it is agitated to form the emulsion. Finally, how you can see in the picture, the ball formation takes place, and finally rigidization of the ball. You get the oil is encapsulated or coated with melamine formaldehyde membrane. The polymerization can be conducted in 3 ways. One is the interfacial polymerization, in situ polymerization, or matrix polymerization. In interfacial polymerization, 2 reactants in a polycondensation meet at an interface and react directly. In the case of in situ polymerization, there is a direct polymerization of single monomer, and this is carried out on the particle surface. Matrix polymerization includes a core material, and this core material is embedded in a polymeric matrix during formation of the particles. It can be done the process either in single emulsion method are in double emulsion method. In the single emulsion method, you can see in this picture, that is IL and polymer, they are mixed using proper sonication or mixing, and water in IL emulsion is formed, and this water in IL emulsion is added with the cross linking agents, all right, and they are heated either by heating, the denaturation occurs, or by the process of chemical cross linking, microspheres are formed in the organic phase, organic solvent, and then once the microspheres are formed, they are isolated either by centrifugation, washing and separation processes. In the double emulsion method, you can see here in this figure, inner phase and dispersed polymer phase, they are mixed together to emulsify it, and in the process water and IL emulsion is formed, first emulsion of water in IL. Okay. And then this water in IL emulsion is added in the continuous phase having surfactant etc., and then again by stirring the second, that is water in IL in water, suspension or emulsion is formed. So, that is the double emulsion, water IL water emulsion is formed, and this finally using appropriate methods of evaporation, heating etc., the solvents are evaporated, and finally the rigid microspheres or microcapsules are found. So, now having studied the microencapsulation processes, another important aspect is the characterization of microencapsules, and there are various methods in the literature which one can use for the characterization. They includes particle size determination or determination of physical properties of the microcapsules, even determination of morphologies. There are various methods available in the literature for these determinations. Even the encapsulation efficiency can be determined, or one can find the viscosity of the emulsion, thermal stability of the microcapsules can be determined, or in vitro methods etc., are available for these determinations. So, means what I try to emphasize here is that it is very important aspects that the microcapsule should be properly characterized, so that to ensure that its purpose, that is the purpose for which the microencapsulation was done, is met with properly. Okay. So, now let us discuss the application of microencapsulation technology in food processing. Friends, you know that several times it is required to add one ingredient into the other in the formulation and processing of different food products, and when many a times these ingredients are not compatible with each other. That is, if you add one ingredient into a food material, it may disturb the sensory characteristics like color, flavor, texture etc., of the original food, right. Also, sometimes the one ingredient which is added into the other food materials or other components, there may be some undesirable interaction between these components, that is the additives added into the food, additives and the food components, there may be undesirable interactions, and which may ultimately lead to the deterioration of the either the original food material, or even reduce the effectiveness of the additives that is added into the. So, all these problems can be taken care of by this microencapsulation technologies, that is the microencapsulation offers promising solutions to these problems in food industry. For example, that you know that EPA or DHA, that is the omega fatty acids which are found in the fish oil, the milk which has more saturated fats. So, this EPA or DHA contents of the milk fat can be increased by adding fish oil into milk. But if fish oil is directly added into the milk, it may interfere with the flavor of the milk may get some sort of fishy flavor, etc. So, the encapsulation of the fish oil provides a good solution, that is, the encapsulated fish oil can be added into the milk without any adverse effect of it on the milk flavor and other characteristics. Similarly, the milk can be fortified with vitamin D entrapped in liposomes. Microencapsulated iron ingredients can prevent off flavor development and maintain bioavailability of the iron compounds. Probiotic bacterial cell encapsulated in calcium alginate are provided protection in fermented frozen dairy products. The objectionable taste and aroma of popular nutritional ingredients like soya extracts, bitter herbs and omega 3 oils, etc., can be successfully masked by using appropriate micro encapsulation process. The micro encapsulations of beta galactosidase in liposome can be used to act in vivo, but it protects from acting on lactose during storage. Similarly, ascorbic acid entrapped in lysosome together with the vitamin E is used for production of emulsion type foods. Even the hygroscopic properties of materials like sodium chlorides, etc., may be reduced by microencapsulation. Fluid bed encapsulated salt is used in meats to prevent the development of rancidity as well as premature set due to myofibrillar binding. Among all existing microencapsulation methods, molecular inclusion of flavor in beta cyclodextrin molecules is the more effective method of protecting the aromas. Even I hope you know that the ascorbic acid is a common additive which is used in bread to improve its quality, but the ascorbic acid, if it is added directly, it can react with the water and oxygen in the dough and gets degraded. But if that is the ascorbic acid is micro encapsulated, then it is protected from the water and oxygen, and therefore, its degradation in the dough is avoided. In this table, this slide as well as in the next slide, I have tried to take material from the literature to give you information on the category of various food ingredients, suitable or preferred methods of their encapsulation and their use in different food processes. For example, the preferred method for encapsulation of acidulants like lactic acid, vitamin C, acetic acid or such other acidulants are the fluidized wet coating or extrusion. These acidulants, that is the coated instruments, are used to assist in the development of color and flavor in meat emulsions, dry sausage products, the meat products containing the other are the meat and meat containing products. They are also used in baking industry. Baking industry use stable acids and baking soda in wet and dry mixes to control the release of carbon dioxide during processing and subsequent baking. The acidulants and other ingredients, flavoring ingredients like citrus oil, mint oils, onion oil, garlic oil, etc. The preferred methods for their encapsulation is inclusion, complexation, extrusion, centrifugal extrusion, spray drying, etc. And these are used to transform liquid flavorings into stable and free flowing powders, which are easier to handle and incorporate in a dry food system. Sweeteners like sugars, nutritive or artificial sugars like aspartame, etc. They are generally microencapsulated using co-crystallization or fluidized bed coating processes, and they are microencapsulated forms of these sweeteners are used to reduce the hygroscopicity, to improve flowability, as well as to prolong sweetness perception. Colorants like annatto, beta carotene, turmeric, etc. They are microencapsulated using extrusion or emulsion methods, and their encapsulated colors are easier to handle and offer improved solubility, stability to oxidation, and control over stratification from dry blends. Lipids like fish oil, linolenic acid, rice bran oil, egg white powders, etc. are spray dried or freeze dried or vacuum dried to encapsulate them, and these encapsulated forms are used to prevent oxidative degradation during processing and storage. Similarly, vitamins and minerals are encapsulated using conservation process or inclusion complexation, spray drying, and such other methods. The micro encapsulated vitamins and minerals like fat soluble vitamins or water soluble vitamins and niacin, folic acids, etc. They are used to reduce off flavors, to permit time release of nutrients, to enhance the stability to extremes in temperature and moisture. They are also used to reduce each nutrient's interactions with other components or other ingredients. Enzymes and microorganisms are microencapsulated generally by conservation method or spray drying method, or even by, and these are used to improve the stability or to reduce the ripening times. The fish oil, as I told you earlier, they are encapsulated, all right, and converted into powder form, and this powdered fish oil is easier to be used in different products. It has acceptable taste and modest shelf life of 3 weeks at around 4 degrees Celsius. Okay. And the raw material for its encapsulation, there is casein in the form of skim milk powder or other such ingredients, and the technology used may be spray drying or vacuum drying, freeze drying, etc. It is successfully incorporated into a number of food products, including infant foods and so on. And in this slides, again different methods of micro encapsulation of fish oil, that is the process flow chart is presented. This is taken from the literature again, where the raw materials are used protein and carbohydrate dispersions, or only protein alone, that is whey protein isolate, or protein plus carbohydrate like soya protein isolate and pectin dispersion. And depending upon the raw materials, they are given the treatment like first the protein and carbohydrate dispersion, the core material used, it is used to adjust the pH to 7.5 and heated to 100 degrees Celsius for 40 minutes. and after this dispersion or emulsion is made, then IL is added into it and homogenized at 350 to 100 bar, and the emulsion thus formed is spray dried at 180 and 80 degree Celsius inlet and outlet temperatures, and the powder is micro encapsulated fish oil powder is obtained, and various combinations and permutations. That is, in one case, that is the emulsion, first emulsion is prepared, first this dispersion is heated and then oil is added, emulsion is prepared. In the other case, the oil was added, emulsion was prepared, and then the emulsion was heated and heated emulsion was spray dryer, and in the other cases, that is the only proteins were used as a wall material, and the effect of all these, that is the process parameters as well as the wall materials, was studied by those researchers on the micro encapsulated fish oil characteristics, and they reported that all these various, they ultimately they give the micro encapsulated fish oil with good stability, good functionality, other characteristics. Okay. So, the micro encapsulation of polyphenol is another important, that is mostly the polyphenols. They have very good antioxidant properties, they have very good functionality, and in many foods many a times they are required to be added, but most of the polyphenols, they have bitter taste or they add. So, when they are added to the food material etc., they impart. So, these polyphenols, if they are encapsulated or and then used in the encapsulated forms, then their interaction with other food components can be avoided. So, from the natural products like grape seed, soya bean, oak etc., the polyphenols may be extracted using standard methods, and then these extracted polyphenols are mixed with wall materials like polysaccharides, proteins, gums etc., to form a solution, and the solution is then, as you can see here in this picture, they are spray dried to form microcapsules. We at IIT Kharagpur have also done some work in for micro encapsulation of edible oils. That is, in our study we used different oil blends like sunflower oil and sesame oil in different proportions, and these proportions are blend were optimized to have high content of PUFA, that is polyunsaturated fatty acids, as well as more antioxidants. So, the PUFA containing oils were micro encapsulated using suitable wall materials like some protein source, some polysaccharide source. This is our patented process. So, I would not like to disclose much the process parameters etc., but you can see here in this picture that is these oils alone or in combination was emulsified with this wall materials and with the suitable aqueous medium, and then oil and water emulsion was formed, and this oil and water emulsion we dried using spray drying as well as microwave drying, and good quality free flowing oil powder was obtained, and these oil powders was characterized, was studied by various, even what is its micro encapsulation efficiency, the amount of oil present in the powder, all those things, and we found of course spray drying yielded a good powder, free flowing powder, and the microwave drying also was found to be equally better process. The oil entrapped in the micro encapsules were comparatively found more in the microwave drying process than that of the spray drying process. We also studied the micro encapsulations of probiotics, because again the probiotic bacteria, it becomes beneficial to encapsulate it and then add it into the food formulation, so that they can perform their activities in a controlled manner. So, we have taken lactic acid bacteria, and again this is also a patented process. So, lactic acid bacteria are mixed with suitable wall materials, that is the sources of the wall materials are maybe carbohydrate sources and maybe maltodextrin etc., or some protein sources, and then in the similar manner they are some aqueous medium is used for their emulsification, and these emulsions are finally, again we have tried two methods, one of spray drying and another freeze drying, and these freeze dried probiotic powder you can see here in this picture, right, and these powders were characterized, their SEM analysis and all those things were done, and we found that in this probiotic powder, that is the activity of the bacteria retains very much, that is its probiotics bacteria. In our study we found that it is highly active, and its the activity even is stored for quite some time. So, after having this study, the microencapsulation process and its use in food industries, the another important aspect is to know the mechanism of controlled release of ingredients, because in this process the bioactives or ingredients, they are coated with certain materials to provide them stability. So, before they are used actually in the food system, it has to be ensured that this coating material is disintegrated, this coating material is ruptured at the time of its use, so that the bioactives or other ingredients are obtained the normal place. So, variety of release mechanism or variety of methods have been proposed for breaking or rupturing of the wall of the microcapsules, and they include pressure or shear stress, melting the wall by suitable method, by thermal method or such other methods, dissolving it under particular conditions as in the case of an enteric drug coating, solvent action, enzyme attack, chemical reaction or hydrolysis or slow disintegration. So, these methods depending upon now type of the microcapsule, type of the wall material, type of the core material, its sensitivity, its strength etc., either of these processes can be used to release the bioactives. You can see here in this picture, like here there is some the bioactives, they are encapsulated, and you see that there is a hard rigid cell outside. So, maybe there is a water or acidic conditions etc., might be these materials, that is they may be water soluble material, they can be dissolved in acidic medium, or depending upon the, as I told you, the characteristics of the wall material, we create suitable conditions is done here, the wall material is dissolved, and finally, the ingredients are released, active ingredients are released. So, this is the control released, means that is all these parameters should be properly studied, it should be properly optimized, so that the where one wants and when one wants these active ingredients to be released, it becomes possible. So, friends, I would now like to conclude with telling you the in brief the overall advantages of microencapsulation technology in food processing. As we have seen earlier, the handling and flow properties of the food materials can be improved by converting liquid to a solid encapsulated forms, the bioactives etc., by their conversion of from this liquid form to solid form, improves their restorability, improves their usability. Even hygroscopic materials can be protected from moisture after being encapsulated. Various properties of active agents may be changed by microencapsulations. In studies on the microphage uptake of microspheres have demonstrated their potential in targeting drugs to pathogens residing intracellularly. Reliable means to deliver the drug to the target site with specificity. The desired concentration can be maintained at the site of interest without untoward effects. The development of cyclodextrin as a wall material has been a major breakthrough in microencapsulation technology. Once the cyclodextrins were made available, it provided a good wall material for improving the shelf life of ingredients or active ingredients. It proved to be very effective in reducing volatility and in providing protection to the heat labile substances. Due to encapsulation, flavors are more stable after processing with microwaves or oven drying or even frying. That is, the micro encapsulated flavors can be heat stable, they can be stable in the heating processes. Liposomes have many benefits for the food industry, including protection of materials until desired released or targeted delivery, etc. So, in nutshell, we can say that this micro encapsulation technology has a very wide ranging application in food processing industries, and it is a very good and efficient method for improving the food functionality and their characteristics and their usabilities, or even for the protecting that is active ingredients from deterioration in the food processes. Thank you.